All right, that's fine. I'll see you all later. Bye-bye.
Yeah, you can do whatever you want to.
So hello, everyone. Good evening and good morning to all the friends who have just joined from United States. Are we live now?
Yes, sir. We are live now.
Okay, wonderful. So thank you so much, everyone, for joining with a wonderful panel and the speakers here. And I would introduce the faculty from United States. We have Dr. Alexandria Rachitskia. She's a faculty at Cleveland. And Dr. Cooperman. He is my mentor, and he's a mentor for Dr. Raja also, who's sitting here with us. And he heads the Gavin Herbert Eye Institute at University of California, Irvine. And apart from that, we have Dr. Pramod Binde, who has the Shankar Nethralaya, a busy hospital in the southern India, and faculty from Disha Eye Hospital, a very busy hospital in the eastern India, and Dr. Alay, who have been associated with biosimilar drugs from the beginning. And he's been quite active with the ASRS too. Apart from that, I think nobody needs introduction for Dr. Vishali. She's at PGI Chandigarh. Dr. Giridhar, who has the Giridhar Eye Institute. Dr. Harsha, he's from Shankar Deva Nethralaya. And Dr. Sarvanan, who is in my same city, and he is from Arvind Eye Hospital. I'm not sure if I'm missing anybody else. Raja, I think we all know from L.V. Prasad Eye Institute, Hyderabad. So the reason behind making it as a Indo-US symposium, probably because has released and launched its first biosimilar of ranibizumab in 2015. So probably we have approximately 5 years of experience that whatever we have gone through. And Lucentis expires its patent in this year. So US FDA has some of the molecules which probably would come into the main streamline this year, or because of the corona, probably the early next year. So we would be having some kind of perspective from Rachitskia and from Dr. Cooperman to understand these molecules in a little more detail. I'm sure Dr. Cooperman, he has a phenomenal experience of pharmacology. He has been associated with all kinds of drugs, and he has seen them with quite close eye. So I think we would be enlightened by his talk also. So without taking much time of everyone, just a single request that everybody should be muted, and if possible, video also to be muted. And we would all be taking all the talks in the beginning. If has panelists or anybody has any question, kindly put it in the chat box, and we would take all the questions during the panel discussion. So after the talk, panel discussion would follow. So I would be starting the first talk, and let me share my slides. So I'm talking about the retinal biosimilars in India, current status and the future perspective. So basically, these are my disclosures. And rather than talking about the current status and the future perspective, let me just go back a little bit. This was the 2015 when India launched the first biosimilar of Lucentis. And it was a kind of a big news, and everybody knew that India was the first one who launched the first biosimilar of ranibizumab. But after launching during the post-marketing time, they had some cases reported of inflammation. Predominantly, inflammation was into the anterior segment, and it was not too many numbers. If you look at the real incidence, it was approximately by 2017, 0.07%, and by 2019, 0.01%. And since 2019, it has been approximately 0.004. So then it was following the similar way the ranibizumab had. If you look at the data, in the initial trials, Marina has also shown some severe uveitis cases, and those cases, some kind of inflammation was keep coming and going. Those reports are there in the post-marketing surveillance data also. Similarly, with aflibercept also, if you look at the retrospective claim analysis, you have severe ocular inflammation reports. So probably, all the biologics, they have gone through the phase where they had inflammation. Second question, if you talk about the biosimilar, is that whether they can display the same kind of potency and the efficacy. So this is the study which I'm sharing. This study was done in 2017. This was by the group of Novartis, Basel, their head office. When they compared these two products head to head, what they found was there were a few differences in terms of few amino acid chains. But if you talk about the potency and the efficacy, it was quite similar. So let me go back and touch the immunogenicity phenomena a little bit, because this is the phenomena that we are trying to understand since last year. And we have published in this area quite a bit, that why these all biologics which we use in the eye have some kind of immunogenic potential. So there are two ways that if you look at it. One is there are some kind of impurities which can come during the processes of this drug. And at the end of the day, these are all drugs which are made in the live organisms. So you have the protein, and protein has an inherent capability of causing antigenicity. So the reason of few batches which caused antigenicity and immunogenicity with the biosimilar was the endotoxin. Because let me touch upon the endotoxin a little bit, which we published. If you really look at the data from FDA has recommendations of endotoxin, limits, probably mostly it as an intraocular use. They do not specify what kind of endotoxin limit you should have for anterior segment or the posterior segment. So that kind of deficiency that we found that, that should be resolved, because if you look at the data from the rabbit model, posterior segment is a little bit more sensitive if you talk about the endotoxin compared to the anterior segment. And that was the reason once the company identified that endotoxin limit is a little high, but then they withdrew the drug and corrected it, and now things are okay. I would just like to have your attention on this particular paper, which is quite close to our heart. This was in 2019, if you look at it. 2019, we proposed a theory, overall theory, that why all the biotherapeutics which are used in intravitreal as an intravitreal injections, and what could be the reasons for the, not having the zero immunogenicity. There is a possibility that you can actually streamline the things at the end of manufacturing, but what would you do with the host? Because ultimately host response, you do not have any control on it. And if you look at this paper, which has now, if you look at the brolucizumab, probably, we are connecting the dots. If you look at the brolucizumab, it has a pre-injection, very high number of ADAs, which we proposed in 2019. Probably ADAs are the ones, which is difficult to get rid of, and they causes probably immunogenicity and sometimes decrease in the efficacy also. Now let me touch upon the usage in India. The usage in India it was 5% if you look at the 2017 data of VRSI. And after a year in 2018, as per my personal communication with the VRSI, 80% of the clinicians, they were quite comfortable in using biosimilar. Now, within a year, there was a drastic change in terms of the usage. If you really look at the reason that why that has happened, probably India was a country which was using a lot of Avastin, and as a physicians, a lot of people were looking at an option where they can feel safe. Why I'm saying that? Because in this country, we do not have compounding pharmacy as robust as you have in the United States. So probably we had a lot of cases of cluster on endophthalmitis, and in between there was a ban on Avastin also, which was released after the effect, a tremendous effort by Vishali, Dr. Vishali and their group. So probably that was the gap which everybody was looking at it, and probably biosimilar has filled that gap in. And as of now, if you talk about the current status of biosimilar, it has been successfully used in all the indications for which we have been using the originator molecules. And these are the studies which are published data in the public domain. Future, what future behold? We have another molecule, biosimilar of ranibizumab, which is going to be launched next year, next month, which is called Reni-Zurel. And probably there are a couple of other molecules, SB11 and the FYP201. When they have this FDA approval, probably then they would be the one which would come into the Indian market. We cannot ignore bevacizumab. We have approximately 6 approved biosimilar to Avastin in this country. And recently, in August 20th, we have an approval of Samsung Bioepis biosimilar in EMA. But I'm not sure, because I do not have any personal experience of using biosimilar Avastin. So during the panel discussion, I think we would discuss this part. So overall, if you look at India, 5 years we have been using the biosimilar. So we have some kind of experience to share to the world. And probably the companies which are in process of FDA approval and trying to launch these drugs, they should concentrate on some of the aspects. For example, immunogenic testing should be really, really robust, because already there is a kind of nocebo phenomena which revolves around these drugs, because they are differentiated from the originator. And on top of it, if some cases of inflammation comes in, then that becomes even worse. Although the efficacy has been great, but even a simple immunogenicity might have an impact on the credibility of the drug. So that is it from the India and whatever experience that we have. And probably this is the year where US would be getting some kind of headlight, in terms of where the FDA approval is going in. And this is the paper that we published in 2018 saying, is there a big change on the horizon with biosimilars in ophthalmology? We saw a big change in this country, and we would look forward from Dr. Rachitskia that what she says, that what kind of perspective US has at this point in time. Thank you so much, and stay safe, everyone. Now I would request Dr. Rachitskia to share her slides. Thank you.
All right. So thank you so much for the invitation to speak. And I'm Alexandra Rachitskaya. I'm from Cleveland Cola Institute. Thank you to Ashish Sharma for inviting me to be among the gurus in biosimilars. So I'm going to give a little bit of the USA perspective. These are my disclosures, none of which are pertinent to the topic of this presentation. So just to give an overview, we all know that the burden of age-related macular degeneration is increasing. This is the projections for United States. And if you look in 2010 at the orange, which is all patients, there was about slightly under 2 million patients affected by AMD. And by 2050, we expect over 5 million patients affected by age-related macular degeneration. In the United States, majority is Caucasian patients, but we see an increase in Hispanic patients as well. And as the numbers of patients with AMD increase, so do the numbers of patients who are going to require injections. So the burden of injection is definitely here, and there's a lot of research happening to minimize this burden. So how do we treat the macular degeneration currently? These are the results of the let me just go back. So these are the results of the Preference and Trend Survey, ASRS, from 2017. And the question was, how do you treat your new wet age-related macular degeneration patient? And it's interesting that in United States, Avastin is the first-line drug for majority of the providers. About 70% of providers start with Avastin, whereas internationally, it's about 30%. And then internationally, we see more Eylea and Lucentis use compared to US. And the question is what drives it? And I think the next question that was asked by Preference and Trends survey is very revealing. And the question is, if Avastin, Lucentis, and Eylea, if the price point was the same, what would you treat your new onset wet AMD patient? And you can see a shift here, whereas in reality, most patients are started on Avastin first. If the price was not an issue, 78% of US providers and 74% of international providers would start with Eylea. And I think it highlights that although in United States, Avastin has been shown to be safe and efficacious, it has a great track record. And as has been alluded to, in majority of cases, we have wonderful compounding pharmacies. Still, it comes to the question of cost. And this is the approximate cost of drugs in the United States. When we look at bevacizumab or Avastin, it's about $50, whereas the brand drugs such as ranibizumab, aflibercept, and the new brolucizumab range anywhere from $1,800 to $2,000. And what happens is it's not only the price of the drug, but also the landscape that US retina specialists function in, because we have a lot of insurance-driven policies of how we treat our patients. We have something called step therapy in some insurances and also in some states where you have to start with a cheaper alternative, which is bevacizumab, and you have to give, for instance, 3 injections before you can ask for approval of the branded drugs. And it gets even more complex because somebody might get approved by insurance, let's say to receive aflibercept, but they have a high deductible, meaning that they have to pay a particular amount until the insurance actually starts covering their medication, or they can have high copays. So in reality, when it comes to my practice, we have patients who are, who are started on Avastin and are capped on Avastin, and patients who also might have great insurance coverage, and we can start them on more branded drugs. But that's the landscape, and I think it's important to understand the landscape and how do biosimilars might fit into that landscape. So just a little bit for everybody to be on the same page, all the drugs that I've mentioned so far are what's called biologics, and they are therapeutic agents that contain proteins that are derived from living organisms. So Avastin, Eylea, Lucentis, Beovu are all biologics, and they require cell line, growth media, post-translational modifications, and purification processes before they're available for us to use in the clinic. And when we talk about the biosimilars, they're biologics as well. And as compared to generic drugs, and I think that's a very common confusion for people who are not steeped into this topic, biosimilars are not copies of existing reference biologic. They're not exact copies. In fact, all they aim to do is to mimic the therapeutic endpoint of the reference biologic. And they're created by reverse engineering work and still require phase 2 trials before they're available for providers. And as I mentioned, all those, even when the patent goes away on the reference biologic, the cell line doesn't become available. So the companies that develop biosimilars have a lot of work to do. They are cheaper to manufacture, and they are easier to get approved than the original biologic. They have to do studies to compare pharmacokinetics of the drugs and the biosimilar of the original biologic and the biosimilar, and the trial to demonstrate clinical equivalence. but cheaper is not necessarily cheap. So here are some of the costs. So if we think about the reference biologics, so all the drugs that I mentioned, the brand drugs, it takes about 10 to 15 years for them to develop. And the cost is anywhere from $1,200 to $2,500 million. So very expensive proposition. The biosimilars, it does take shorter time for them to develop, 8 to 10 years, but the cost is still very high. It's $100 to $200 million. Whereas when we think about the generic drugs, the time to develop is 3 to 5 years, and the cost is somewhere between 1 and 5 million. So looking at these numbers and the resources that need to be put in, both in terms of the capital and time investment, you're not going to have small companies coming into this market because it is quite expensive. And if you invest all this time and effort and money, then the important question, and it goes back to our landscape that we practice in the United States, where are biologics going to fit in? What is going to be their price point? So we have currently a great alternative that is cheap with bevacizumab, and the biologics, given how the biosimilars, given how expensive it is to develop them, there's no way they're going to be close to the cost of Avastin in the United States. And also, as I mentioned, we have then the branded drugs that a lot of patients, their insurance cover it. And then the question is, if there's a biologic, are the branded drugs going to decrease their price to be competitive? And now we have a drug that has been around for a while. We know their efficacy, we know their safety, and how is that going to compete with the biosimilar? So the manufacturers of biosimilars will have to market their products and negotiate individual contracts with purchasers to be competitive in this market. The other important point that has been brought up is the safety. And we all are very concerned about safety, especially in light of some of the new reports with the medications that are in trials and also recently approved, as you mentioned, with Beovu. And I think all the providers are very concerned about intraocular inflammation, as well as more significant safety concerns with occlusive vasculitis. So once again, now I think bringing a medication to a market, you have to show really robust safety data. So in summary, I think in the United States, there's definitely a potential niche for biosimilars. But they have to be considered in the current context of a variety of other options available and the medications that are currently on the market. And I thank you for the opportunity, and I thank you for your attention and look forward to the panel.
Thank you so much, Dr. Leticia, for giving a wonderful perspective from US. Now I would request Dr. Cooperman to share his slides, please.
Thank you very much, Ashish, for the invitation, and Alexandra, great presentation. I'm going to overlap a little bit with it, but that'll be fine. So again, I'll be discussing the originator versus the biosimilar drugs, key differences, and I want to acknowledge both Ashish and Raja, who were former fellows of mine and have been wonderful collaborators. We've collaborated across our continents and have written many papers together. And then you've noticed the papers that Ashish referenced that I've been involved with, and also Nilesh Kumar has been such a productive writer as well. So thank you for the collaboration. It's been a joy. So I will continue now by way of, let's see, it's not, there we go. That's by way of disclosure. I consult for a lot of companies, but none are relevant to therapies, except of course the companies that will end up being competitors to future biosimilars. So what are originator molecules? I'll discuss. What is a biosimilar? What's the need for a biosimilar? And how is a biosimilar different from originator molecules? So the originator molecule is the biological innovator molecule, the ones that we all use, as was discussed by Alexandra, to which a biotherapeutic drug indication is being developed. It's typically a single therapeutic product authorized and regulated to be used in the market. And if it's associated with the development of a biosimilar, why do biosimilars even exist? It's because the originator molecules are safe and effective. We've learned to love them and use them. It's the bevacizumabs, the ranibizumabs, the aflibercepts, and others. But the biosimilars come along because there's patents. And patent is, as it begins to end, then the competition begins to think, can we get a piece of that marketplace? But of course, patents themselves are not simple things. They vary over the course of the world. The patents applications have durability that's different around the world, and the applications were made at different times. So there's no such thing as a single patent expiry date. But the bottom line is, for the big three that we all use, between bevacizumab, ranibizumab, and aflibercept, all the patents are either expired or expiring as we speak. And by, I think, around the world by 2025, all the patents, the last one to be have patent expiry will be aflibercept. And again, as it's already expiring in the United States and will be expired by 2025 at the last part of the world. Again, it's a huge cost, as what's been highlighted by Alexandra. The global market for ophthalmic drugs is $31 billion US dollars. Anti-VEGFs occupy shocking 11.6 billion, 37.5 of the global market. The United States accounts for a little over half of the global anti-VEGFs revenue, which is an interesting phenomenon if you think about it. You saw how popular bevacizumab is in the United States. It dominates the United States because of its low cost in the eye. Of course, when we say bevacizumab is inexpensive, that big bottle of bevacizumab that's used systemically for infusion is something like $4,000, but we can take that little bit out, and we're allowed to aliquot it essentially from the compounding pharmacies, and it costs roughly $50 compared to the roughly $2,000 per dose of ranibizumab and aflibercept. So again, imagine how much the United States would be spending if it was paying, if it was not using Avastin, bevacizumab, and it was using just the premium products, as we call them. The developing nations have a huge unmet need with poor universal health care coverage, out-of-pocket expenses, lower per capita income. And of course, ironically and sadly, United States, the richest country, has the ability to use this compounding pharmacy and bevacizumab, Avastin, but because of the complications, the risk, the contamination risk, et cetera, many poor countries around the world really were shut down and do not have access to this form of aliquoting of compounding the bevacizumab, so they don't have that inexpensive option available to them. So what's the answer to all this? Biosimilars in ophthalmology. Is there a big change on the horizon? Ashish referenced this, and again, I leave it here so that you can look at that paper, but there's a lot of papers that we sort of discussed what's going on in the future, but a lot of this, again, is the patent expiry, and so we anticipate by 2025, there'll be the biosimilar option available around the world. As Alexandra very properly said, will it succeed or not? And again, there's a lot of concerns, but I'll get to that as I go through my talk. What is a biosimilar molecule? We've heard that already a bit, but they're equivalent to their originators in terms of quality, safety, and efficacy, but they're not the same molecule. These are not generics. These are not simple to make. These are biologics, as has been mentioned. They come out of E. coli or hamster ovary cells or wherever they come from, wherever they're generated, and they can have host cell impurities. They can produce inflammation. So safety is a key element, but also, of course, efficacy, because these are not the identical molecule. They share similar features. They have to have the same receptors, the same efficacy points, but it must prove similar pharmacokinetics, pharmacodynamics, immunogenicity, safety, and efficacy to the innovator originator biologic molecule. So this is not a trivial thing at all. It is creating a new molecule. It's reverse engineering, as the term has been used. It is definitely not a generic, night and day. Because of their complexity, which includes natural variability, they're strictly separate from generics. It is extremely different from generic, but one of the battles that we have to fight when we do roll out our biosimilars is that patients may worry that they are generic and may be cheap and not as good. So that'll be part of the issue, which I'll address momentarily. Again, they have to be similar to the originator molecule with high analytical similarity, the same amino acid sequence and folding, highly similar analytical profiles based on these sensitive methods, comparable stability profile, closely matching pharmacological actions, and similar purity. And purity is probably where brolucizumab and another compound, abicipar, by Allergan, that had inflammation, may have been, at least abicipar very clearly identified their main concern with host cell impurities. So again, even originator molecules have to have very high purity. And we've been very fortunate that bevacizumab, ranibizumab, and aflibercept are not only very effective, but they're very safe. And I think now in this era of brolucizumab and abicipar, that we're going to be the new generation of Q12 dosing, every 12-week quarterly dosing. Unfortunately, that is being put to bed for now until they can sort out the problems, but inflammation was the issue with those new originator molecules. The biosimilar similarity to originator molecule has to have a confirmation of biosimilarity in the pharmacokinetic pharmacodynamic study and or clinical study. They have to have comparable human PK and pharmacodynamic profiles, and again, comparable safety and immunogenicity. And they have to find an immunocompetent population for studies to study the immune reactions with high sensitivity. They're very different from the biosimilars are different from the originators. And again, there's an article here that Ashish is the lead with Nilesh as well, having a role in understanding biosimilars and its regulatory aspects across the globe, an ophthalmology perspective. And again, there's the rules are different for each country. There's certainly, as has been seen from Alexandra, they're very different. The investment for a generic is very different from a biosimilar. The investment for a generic is $3 million. An originator is $1 billion, shall we say, $1,000 million to $2,000 million. And biosimilars are a fraction of that, but still much more than a generic cost. The time to market is, as Alexandra mentioned, for a biosimilar is less than the originator. Number of patients are needed is much less than originator, but compare it to the generics, much more difficult to develop a biosimilar than a generic. And again, you can see some of the differences here. Generics have simple and identical structure to the molecule. So, and all they're looking for is once they show that similarity and they've got a vehicle that seems safe, the generic can be approved without really testing it in humans, whereas biosimilars have to go through an arduous process that is akin to a originator, but just not as extensive. Again, the originator versus biosimilar, as I show here, you have to do the complex analytics. You have to have phase 1 trials are extensive. The dose should be similar, and there are indications that are tried to demonstrate clinical efficacy, and then they're extrapolated. So you don't have to prove every indication with a biosimilar the way you do with an originator, but you do need to show it for at least 1 or 2. Again, and to conclude, there's now a new era of, and again, an interesting paper that I reference here, where it's not just biologics and biosimilars, biologics being the originator molecules, the biosimilars being the follow-ons that are cheaper. But now in this technology, once you identify something that is good and you know how to make it, the reverse engineering of biosimilars, there's actually now something called bio-betters, where you think you might have an ability to modify that further, this reverse engineering of taking the good and making it better, the realm of bio-betters. But it's interesting that a bio-better, because it is different now biologically, structurally, to some degree in the amino acid sequence, et cetera, than the original originator molecule, is viewed as a new drug, and so has to go through the full process of an originator molecule. They're cheaper to develop because you're standing on the shoulder of a giant, so to speak. So you're reaching higher up, but you still have to go through a full approval process with the multiple levels of drug trials, but your chances of success are higher because you already know that the foundation molecule has been shown to be safe and effective. But again, safety is a key word with this as we go forward. So the future depends on education of physicians, counteracting the nocebo effect. Again, this notion that patients may assume or fear that a biosimilar is not as safe or effective as the originator molecule and is just a cheap generic version of the originator molecule, that is a concern that we have. I think we can allay that, but we're going to have to counsel our patients. The availability and pricing of biosimilars, will they be competitive enough, will be low enough to accept that trade-off from a molecule we're used to. And also, finally, the biosimilars to the aflibercepts, to the bevacizumabs, to the ranibizumabs, as they come on, are we going to start shifting to a new generation of drugs? There's the ranibizumab port delivery system with new molecules with potentially superior efficacy or durability. There's faricimab, which again, that we thought that would be brolucizumab. We thought it would be abicipar. Those aren't online, but we're also going to see Combercept, new molecules coming along that will ultimately be, will have their own patent expiries and may have increased efficacy that may alter this landscape as well. So I'll pass the baton. Thank you very much for the invitation, Ashish. Thank you all for listening, and you're all welcome to come visit at the Gavin Herbert Eye Institute at the University of California, Irvine, where Ashish and Raja spent a couple happy years with me, though I don't think we had this building built then. That was back in the old days. Thanks again for your attention.
Thank you, Dr. Cooperman. I think you rightly said a couple of happy years. I think both would definitely agree with that. So with that, I would invite Dr. Pramod Dr. Pramod he has the Shankar Netralaya. I think that doesn't need an introduction. So he will share his experience.
Yeah, good evening, everybody, and good morning, Dr. Kupperman and Alexander. So, can you see the slides?
Yes, very well.
Yeah, so as outset, Ashish, thank you very much for giving me opportunity, and congratulations for getting things organized. My greetings to Madhusudan as well. When I'm seeing him, so happy to see you once again. So basically, just to overview of what guidelines we follow when you give intraarterial injection at Shankar Netralaya. Basically, all the injection are given in operation theater, like taking all precautions with what any any surgery, ophthalmic surgery we do, like cap, mask, speculum, proper draping, as you see here, and using a microscope. Patient is properly draped, surgeon is having a gown, glove. We don't use a pre-injection antibiotic, but ensure povidone iodine 5 to 10 minutes of contact time before injection. And once injection is done, the patch is kept for 2 hours after using again povidone iodine, and patient goes back home. All these patients, as you say, we ask them to avoid head bath for a couple of days, and clear instructions are given about possible signs and symptoms related to emergency. We see the patient next day, again complete examination including dilated fundus evaluation. However, we have changed the protocol recently because of COVID period to minimize the contacts. We just all start a teleconsultation for this patient, and if patient is symptomatic, then only he comes back for a checkup. And as usual, the standard protocol, 1 month review, again complete examination, OCT, and depending on the findings, we decide about subsequent injections. So we had initially hesitancy to initiate use of Razumab. Shankar Netralaya was not the part of multicenter trial which was done by Intas, but when we wanted to start at exactly that time, we had all this injection inflammation due to its polysorbitate content, and that compounded the matter. Finally, we decided to have our own pilot study to look for safety of injection after the drug, which got reintroduced after changing that polysorbate, and what Ashish has rightly described already. So our aim was to look at the safety profile of Razumab and to look for efficacy as well, though we knew it's a similar molecule, so efficacy probably should not be an issue. So it was a 2-month trial in 2016, which 31 injections we gave. 27 patients were followed for 1 month, and then 8 patients for 2 months, and then majority were diabetic, and the second one as AMD. So when you look at it, visual acuity, yes, you can see more or less in all indications, definitely visual acuity has improved both at 4 to 6 weeks follow-up and also 8 to 10 week follow-up majority situation. But when you see here as a OCT thickness, this is a typical again anti-VEGF graph we see at 4 to 6 weeks, thickness has gone down, but again by 8 to 10 weeks, it started picking up as well, because the effect is like a drug is gone now. As far as IOP is concerned, there's not really significant alarming IOP change in most of these eyes except C and VM group. Probably you see it's compared to baseline IOP was higher, but it's still in an acceptable range, particularly when 8 to 10 weeks pattern as you see here. And same continues. And these are just representative cases. That time the good effect just showing why this is a branch vein occlusion, another branch vein occlusion come down from 540 to 260 as a central field thickness. This is a diabetic macular edema. SRF is now gone when only 1 injections from 564 to 168 thickness has reduced. This is another diabetic, though thickness has gone around, but still there is significant amount of thickness is there, but subretinal fluid got absorbed. And this is another CNVM patients, again, similar effect showing good efficacy. So basically of these 31 injections, 1 was doubtful, but 1 patient definitely had antechamber reaction at day 1, but it resolved completely on day 3 without any specific treatment, and we did not have any other serious adverse event. So we concluded that yes, we have a good safety outcome, and as far as short safe term efficacy is concerned, definitely response is good. These are just, as I said, only 1 single injection response what we have seen here. That was our first study, And then we decided and open up that we'll go ahead with injection, open for all the patients. And then another subsequent group we analyzed between 2016 and 2018, 2 years, 190 injections. Treatment naive patients were 148. I'm just till today, we have more than 2,300 injections, Razumac injections were used. That data we need to analyze further again. But with this 190 patients, of which 140 treatment naive, when you look at it, look at visual acuity and central foveal thickness, which definitely visual acuity is better. Average, you look at it, thickness has gone down, and IOP, there is not much change. When subgroup analysis look at a diabetic macular edema, again improved visual acuity, definitely reduced central foveal thickness, is no effect on intraocular pressure. Look at a venous occlusal, similar response, no IOP changes, so no cause for concern, 37 eyes. When you have and you have a CNVM, numbers were very less in that group. Again, visual acuity, again, because visual acuity here obviously was affected because of location of the lesion and probably previous marginal scarring, but definitely thickness has gone down. Again, like previous study, a small short report we had, experience we had, IOP is marginally higher, specifically in CNVM group, though number is too small to really conclude anything, and we need to analyze it further. But definitely this is the only group we have IOP is marginally, though it is still in acceptable range. We'll look at it. So we concluded here, yes, Razumab is safe and effective. And as Ashish, again, his presentation said that yes, it is approved in most of the common indications. The advantage is it's a single-use vial, so no problem of fractionating or pooling of the patient. It is cost-effective and affordable, and results match to the innovator drug. So this is what our conclusion, and just Ashish wanted me to have some report, typical cases, not exactly typical, rather I would say some little bit unique cases, little bit difference we have here. This is a 50-year-old male, diabetic, hypertensive, had CNVM in the left eye, received 12 Biosimilar and 1 ranibizumab between 2012 and 15. Then he have a quiescent period of almost 2 years, and then reported back with reactivation in February 2018. This is what the picture here, reactivation. So first injection was given in February, had a monthly injection like March, then subsequently March end, May, early May, June, July. And July, because we thought it's a little bit better, vision also has improved. There was suspected lesion here, but we thought let us observe. So we call him, instead of giving injection, call him again after 3 weeks. And then, but it's worsening, so we decided go ahead with another injection. Again, 1 month gone, and subsequently we'll continue. And then you see January, April, and what happened here basically wanted to show he probably sort of settled down in a pattern between 3 to 4 months repeat injections. And that is what we all know. This is what we are experiencing now. So the patient was treated with 4 monthly injection and followed by PRN basis and had good response. So, but injection needed to be continued. None of the same sitting, none of the injection, we did not have any adverse effect during this case. Another case, 74-year male, distortion of object, vision was exceptionally good, normal 6.6 and 6. You see this picture again, had a 5 monthly injections, and sort of he settled down, then vision is maintained at 7.5. We have a continuous follow-up till May 19, and then he come back with another recurrence, settle down, 4 months gap, nice maintain vision, again come back with recurrence, again 2 to 3 months gap, another recurrence, we have to re-inject. Again, observation in between, and another injection, that is June 2020 now, the last injection we gave, and he is yet to, we are waiting for his follow-up now. So 9 injections here. Again, it proved that just drug is effective, it is safe, but recurrences are common, and both this first and second case shows that over a period of time, all these patients may settle down in their specific treatment regimen, probably pattern and interval at which they need injections, and we probably learn over a period of time and then accordingly follow these patients. This is another lady, 55-year-old, had a cataract surgery. Initially, everybody thought it's like a post-cataract surgery. CME was treated with bromfenac. Vision was good, but when followed over almost a year, in May 2018, realized vision is not, in fact, gone down further. So thought of diabetic CME now, because initial was confusion. So she was given option of Ozurdex versus anti-VEGF. She opted for Razumab for second injection. There was initial response, as you see, but subsequently, though vision has improved, but look at macular edema, probably almost persistent. There's not much difference. So we gave another option in February month. Shall we switch to another anti-VEGF, or whether we should have Ozurdex? Somehow she was not keen to have Ozurdex, decided to have. Now here is Accentrix we gave. And again, look at this, not much difference as far as edema is concerned. Vision also now August has gone down. Somehow we convince her to change to Dex, now Ozurdex, and with one injection, excellent response we got for her. So had a chronic DME, as I said, not responded well to anti-VEGFs, but have a good response to dexamethasone. To complete the story, she came back again in January 2020, recurrent CME now because of the gap of 4 or 5 months. She refused the injection now. Again, June 2020 DME. Now she has both eyes similar problem, but she agreed to have injection only in one eye. And somehow we just left her with that injection. Another patient, non-diabetic, CME here, otherwise NPDR. Both eyes similar problem. Multiple injections going on, edema is more or less, initial response was there, as you see, progressively edema going down. Fortunately, vision throughout this follow-up, multiple injections are good. Here we decided to observe, but then he came back with based on FFA done outside. We did a focal to macroaneurysms as well, but as you see here with the subsequent injection and even again changing to Accentrix, again the same scenario, we again gave him options of having it steroids here, patient did not want, and exactly similar response having in other eye as well. Only thing is here, patient refused to have steroid, again wanted to have Razumab, October 19, this is a last follow-up. So what we learn here, yes, there is an initial good response. Probably patient have developed resistance, but if there is no response with one anti-VEGF, probably switching to another anti-VEGF may not benefit here. And probably I feel steroid would have been a better option in this scenario. So another case, similar, if you have a tributary occlusion, we advise anti-VEGF, patient refused. But what happened here is came back with a worsening of the situation, though vision was reasonably okay, but thickness has increased, had a 4 Razumabs here, monthly probably, did not respond, then switched to Biosimilar and see the one injection, vision has improved, macular contour is back to normal, but again, another recurrence here. Because it's September, October, 1 month effect is gone, though vision is maintained. So patient was lost to follow up till he came back, but had another episode of vascular occlusion now, and thickness almost 758, though bevacizumab is reduced. But again, 405, decided to give him dexamethasone at that time. When you see July and January, you have a thickness has increased again. Probably what happened here, what I look at it, yes, there was no response to Razumab, had only initial response to bevacizumab. Whether you call it no response to Ozurdex, but probably had an initial response. But when you look at here, almost a 6-7 months gap, have an initial response, and probably what we are seeing is recurrence because in between patient did not come back for follow. So that also contributes. So I cannot use the word exactly no response to Ozurdex. Probably repeated injections would have helped him. And this is the last case, single injection, very good response for a tributary occlusion, but came back with recurrence. Typically what you see, fortunately, this patient throughout maintained 6/6 vision and multiple injections, again, changed to bevacizumab. We were observing, but though vision is maintained throughout, macular edema also was there. He is being followed up. The last injection almost 1.5 years back, vision is still 6/6, macular edema still there. Now, when you look at this case, there's not really much response either to Razumab or Biosimilars. I do not know whether steroid would have been better in this scenario. We have not tried this, but it again there is the question what we always have: 6/6 vision with chronic CME. Do we really need to treat or not? I've just leave that question open here. So, but he's still after almost 2 years follow up and still maintaining 6/6 vision with that residual edema. So, thank you very much. I'll stop here. Thank you, Ashish, and thanks for patient hearing.
Thank you so much, Dr. Binde, for sharing all these wonderful cases. And I would request now Dr. Debulal to share his slides, please. Dr. Debulal has rich experience with biosimilar. He's working as in quite a busy hospital. Over to you, Dr. Debulal.
Thank you, Dr. Ashish. My slides are visible?
Very much.
Okay. So very good evening and good morning to everybody, depending on where you are at this point of time. So I'll be speaking on biosimilar ranibizumab or Rezumab in wet AMD, the Disha Eye Hospital story. I have no financial disclosures here. So the shift of global healthcare from small molecules to biologics is hard to ignore, as we have heard the previous speakers mention. And the problem that the biologic drugs bring forth are in terms of drug pricing and affordability, especially in this part of the world. So the role of biosimilars have never been more important. So India today has a robust platform of biosimilars in various stages of usage and development. And coming to the eye or ophthalmology, it was in 2015 when, as Dr. Ashish has mentioned, we had approval for the biosimilar ranibizumab for intraocular use. So let's go a little back in time and see what the Disha Eye Hospital story regarding anti-VEGFs is all about. So in 2015, we were actually giving around 15,000 injections, and this was, of course, Avastin, Lucentis, and some IVTA. But at that point of time, we had serious issues with bevacizumab, and we had to stop using bevacizumab or Avastin from 2015 onwards. So next year, the story was like this. We actually went down to 6,500 injections, or roughly half injections, with no Avastin being used. Then we were actually on the lookout for something that would be cost effective and would be useful. And then we started using the biosimilar ranibizumab. And in these years, we have moved up marginally, but as you can notice, we haven't really reached the standards or the numbers we had when we had Avastin in usage. So last year, we injected a total of 9,500 odd injections. And in these years, starting from 2016 onwards, we've used a total of 8,500 odd biosimilar ranibizumab or Rezumab injections. Now, as Dr. Pramod Bhende sir just mentioned, we all have experience regarding the effectiveness of anti-VEGF. So Rezumab or biosimilar ranibizumab, being an anti-VEGF, is likely to be effective. So we have experience, most of us have used biosimilars by now in India. And so we have experience with, these classic CNVMs. They, do good over a period of time. This patient, for example, had 6 injections from 2018 onwards. And for the last 6 months is off any anti-VEGF injections. But let's look at this patient. This is a 67-year-old lady who initially presented with decreased vision in the left eye. The right eye was already in a terrible state with 360 in the right eye and 624 in the left eye. And as you can notice, there was a RP rip here, sub-RP fluid and subretinal fluid also. She was offered a bouquet choice of anti-VEGFs, aflibercept, the innovator ranibizumab, and of course the biosimilar ranibizumab also. So considering all the expenses and everything, she opted for the innovator molecule, the innovator ranibizumab molecule initially, and she started doing better. The subretinal fluid gradually decreased, but the sub-RP fluid was there. And this was 2017. So rest of 2017, she gradually started improving, moved up to 612 from 624. And this was early part of 2018. Rest of 2018 also, she had a waxing and waning subretinal fluid. The sub-RP fluid was more or less similar in 2018. And then she disappeared for a while and came back in April of 2019 with this subretinal bleed here. And of course, the vision had gone down. And this time she said, I will not be able to afford the innovator molecule. And she moved on to the biosimilar ranibizumab. And from then on, she again started improving, the vision again started improving. The subretinal fluid here is gone, but the sub-RP fluid is as it was. And this is how she fared through rest of 2019. She is roughly on 5th week or 6th week biosimilar ranibizumab injections, and her subretinal fluid keeps waxing and waning. And this is just a couple of days back. And she has received another injection of a biosimilar ranibizumab. So all in total, she has received 21 injections so far between the innovator and the biosimilar drug. But moving on to our experience, the entire experience with treatment-naive neovascular AMD, we have this study. This was non-randomized, retrospective, open-label, multi-center phase 4 study of 243 eyes done across various centers of Disha hospitals, 15 centers to be precise. And we looked into the change in best-corrected visual acuity between baseline and final follow-up. And the secondary endpoint was the number of intravitreal injections that the patient received. And we also looked into safety to observe any anterior chamber or posterior chamber intraocular inflammation. So these were, of course, treatment-naive patients. Each eye could be included as separate case, and at least 6 months of follow-up was necessary. No switching between drugs were allowed. No patients who had been treated earlier with any other anti-VEGF was allowed, and any other coexisting retinal disease or surgery, the patient had to be omitted from analysis. And these were the results. They were a total, as I mentioned, of 243 eyes with the mean age of 66 years. This was between June 2016 to February 2020. This is the distribution across different years of usage. And if you look at this slide, the patients who actually stuck on to the same molecule for 6 months of treatment was 85%. But from then onwards, the numbers actually start dwindling. So between 6 and 12 months, they were roughly 10% of the patients who stuck on to the treatment with injections of biosimilar ranibizumab, and beyond 12 months, it was roughly 4.5%. Now, what about the baseline visual acuity? Well, a majority of the patients actually had between 360 to 618 visual acuity when they initially presented and received treatment. And if you look at change in best-corrected visual acuity across all eyes, they were around roughly 20% of the patients who actually went on to have decreased visual acuity. 27% had unchanged visual acuity, but the silver lining was that more than half of the patient, 52% roughly, had increased visual acuity. So if you look at the number of lines of improvement, less than 1 line, 4.9%, 1 line or more of improvement, 40%, 2 lines or more, 31%, and 3 lines or more, 23% patients. If you talk about across all visual acuities, then the change in the visual acuity from baseline of 0.89 logMAR units was to 0.75 logMAR units. The change here was 0.14 logMAR units, which was statistically significant, as also across other visual acuity baselines where the visual acuity improvement was similar, and this was statistically significant. And if you look at the number of injections, the mean was 2.38. And if you consider 3 or more injections, roughly 25% of the patients in this cohort had 3 or more injections of biosimilar ranibizumab. So what about serious adverse events? Well, one patient in the course of treatment, 3 weeks post-injection, had CVA. Don't really know whether we can associate it with the injection itself. Our patient was 81 years old. And regarding intraocular inflammation, 2 eyes had anterior chamber cells. We didn't have to use any intravitreal injections for this. Topical steroids were sufficed, and the patient, the anterior chamber inflammation resolved in 7 days' time. So to conclude, biosimilar ranibizumab shows the desirable qualities of ocular anti-VEGF, and retrospective observational data shows effectiveness and safety. However, it is very important to maintain very high reproducible standards of manufacturing in order to sustain the high drug quality that has been established. So I'd like to end with this slide. Between 2016 and 2019, actually at Disha Eye Hospitals, we have advised a total of 46,000 odd anti-VEGF injections, out of which only 52%, that is 23,000 patients, actually went on to receive an anti-VEGF. So the question here is, did 48% of the patients could not actually afford an anti-VEGF injection? So this is something that's very important. We need to think, and the challenge ahead is to provide a safe and cost-effective drug to a larger population. Thank you for your kind attention.
Thank you, Dr. Devular, for sharing such a rich data. And now I would request Dr. Alay to present his work on ROP.
Thank you, Ashish, for this kind invitation and allowing me to share a few cases of my experience with using this biosimilar in ROP. I have no financial interest, and of course, I'll be presenting the off-label use of this drug in ROP. As you've been hearing, and you all know, I'm not going into the details, but anti-VEGF in ROP is a very debatable topic, with the BEAT ROP study mentioning about bevacizumab, and recently the Rainbow study showing the efficacy of ranibizumab. We've heard from previous speakers about the Avastin ban in India in 2016. And it was this ban that gave us the first opportunity to use the biosimilar drug in ROP. So I'm just going to show a few cases. That is what Dr. Ashish has asked me to do. This is the first case. This was a baby who was 8 weeks and 4 days preterm with a birth weight of 1,300 grams. And an NICU stay, pretty long NICU stay. And this baby presented with severe form of APROP. And what in India, we are calling this as a half zone ROP, where you can see the blood vessels not even reaching, just touching the fovea, not even going beyond the half meridian. So this is what a very severe disease. This is the right eye and the left eye also. There is only a peripapillary vasculature in both the eyes, hardly any vessels reaching up to the fovea. So if you see, we went on to use Razumab injection in both the eyes. And this is 12 days post-injection. You can see the regression of the plus disease and the tortuosity dilated vessels. The vessels have now started progressing into the periphery. A few retinal hemorrhages are still seen. But you can see this picture, it looks like a fairly normal looking eye as compared to the previous angry looking and incomplete eye, as we call it. This is 1 month post injection. You can see the vascularity is now progressing onto the periphery very nicely. This is 2 months. Again, you can see the vasculature now has gone beyond zone 2A into the zone 3. In both the eyes, you can see very nice vasculature developing right up to the periphery, up to zone 3. That is 2 months down the line. And this is just one injection. This is again 3 months. This to see how beautiful the vascularity looks like. It's almost impossible to believe what the vasculature was at the baseline. And this is almost a total vasculature with an extremely narrow peripheral avascular area in zone 3. So just to summarize, basically, this is the presenting picture, and this is at 6 months down the line, almost complete vascularization. You can see how beautifully it looks like almost a normal retina. And we went on to receive the very thin strip of the peripheral avascular retina in zone 3. And this is our protocol. We usually follow them up till 50 to 60 weeks. And only if we feel then, we add on laser later on after about 60 weeks. This is another interesting case, a case of zone 3, zone 2 posterior with plus disease who had already received eccentric injection elsewhere in October 2018, had a very, very bad preterm history. And this is 2 months post the eccentric injection, which we had received elsewhere. And this is the picture on presentation to us. And you can see the looping of the vessels just in posterior zone 2. There is, you can see this watershed layer of the retina, which is just developed here, almost zone 1, not beyond that. This is the right eye, and you can see the similar picture in the left eye. And we went on to give the Razumab injection, and you can see how just in 9 days, you can see how beautifully the entire disease regresses, and the vasculature now, the looping is gone, and the vessels have started progressing into the periphery. And this is 4 months post-injection. You can see the vasculature reaching almost up to zone 3. This is the left eye, and you can see the right eye also, the vasculature reaching almost till zone 3. We then follow up our patients on indirect ophthalmoscopy, and this is the patient 7.5 months down the line on indirect ophthalmoscopy showed complete vascularity up till 1 disc area, which we actually term as fully vascularized retina, and the patient did not go on to receive any laser. This is another case, again, an 8-week preterm. This is actually a larger baby. You can see the 2,250 gram baby had sepsis, apnea, phototherapy, neonatal jaundice, but presented with a very bad disease, a stage 3 zone 2 with plus disease. You can see the left eye, very severe plus disease. When we give the injection, you can see in just 1 week, the entire plus disease completely regresses. And you can see the normal looking vasculature, and now the vascularity is progressing up to almost zone 2 anterior, even in 1 week. This is 5 months. You can see it's almost fully vascularized, very nice. Just this temporal zone 3 is still avascular in both the eyes. You can see this patch, it is avascular zone 3 area. And we went on to follow up this, and the vascularization actually went on beyond the ridge, and the patient on last follow-up had fully vascularized retina with a very mild myopic refraction. This is again another case, a 1600 grammer, 12-week preterm. You can see patients with both eyes, APROP, also known as hybrid ROP, with Dr. Dogra and PG as described. You can see this dirty looking grayish membrane in the peripapillary region with marked plus disease in both the eyes. And just 1 month after one injection, you can see how nicely the disease regresses. And there is still a large area of avascular retina beyond zone 2 anterior. So if you see these patients further down 2 months, now it has reached up to zone 3. There is again an avascular patch in zone 3. And we have a complete avascularity, almost up till 1 disc area with almost 9 months follow-up. And this is 1 year where the refraction was done with mild myopic refraction in both eyes. So this is the compilation of the data. You can see the initial pictures, and this is the final picture. The patient still has a narrow zone of avascularity, but we are still following up the patient. Another patient, a very, very low birth weight baby, 700 grammer. And you can see how severe the APROP was. You can see very severe disease, almost in zone 1. And if you see after 1 week, there is complete regression of the plus disease. And you can see the beautiful demarcation line between the vascular and avascular retina, but see the caliber of the vessels returning almost to normal. And if you go on seeing these patients, that is 1 month, it has reached zone 2A. And then we went on to do the laser because the vascularity stopped progressing, and we went on to give peripheral laser. Why are we discussing the importance of anti-VEGF in these kind of diseases, particularly the APROP or zone 1 or zone 2 posterior disease? And this is a research that we just presented at this year's ASRS. This was one of the babies initially where we used to do laser in APROP. And this is the baby now. She's almost 13 years old, and you can see how the fundus looks like. And what we are trying to study is the effect of the visual fields. And this is the visual field of that girl. It's completely destroyed, only a tunnel vision. And as opposed to this, this is one of our very early cases way back in 2008. You can see the APROP, which was treated primarily and only with anti-VEGF without laser. This is 12 years into follow-up. You can see her wide field photography, complete vascularity beyond zone 3. And these are the fields. So you can see the effect of the anti-VEGF without laser, that there is a very good preservation of the peripheral visual fields. So thank you very much for a patient hearing. And I would like to thank Ashish for giving me this opportunity to share my experiences.
Thank you so much, Dr. Alai. I think it was a wonderful experience, and I already have some questions for you. I think now all the panelists can be seen, and I think we can start. We have sufficient time to discuss. So, Dr. Alai, I think Dr. Subhadra, she is just asking a question that's regarding the dose of Resumab that you use, whatever cases that you have shown. Subhadra, Dr. Subhadra, if you are hearing, as regards to you.
Yeah, for whether it is the biosimilar ranibizumab or the Eccentrix ranibizumab, I always use the full adult dose. The main reason why I do is I personally, with my experience, still believe that the wow effect that we see after the anti-VEGF injections in ROP after Avastin use is not as much with the ranibizumab, whether it is the Novartis ranibizumab or the biosimilar. So I tend to use the full adult dose even in the premature babies. I used to use the half the adult dose, but I used to see a delayed response and also recurrences almost within 3 to 4 weeks after mainly eccentrics. So this has prompted me to use the full adult dose.
Dr. Pramod, do you have anything to add? You see,
Yeah, I am still happy, like most of our cases, various reasons, I would still prefer Avastin for ROP.
Yes.
Okay. And yes, Razumab and Eccentrics, what we use, we have experience, and what Ali said, I totally agree with you. You get a recurrence, is even we have seen recurrence as early as 2 weeks, between 2 and 3 weeks. But I'm hesitant to use a full dose. I always use half dose and probably follow these patients closely. And maybe by 3rd week, you're already having somewhere anterior zone 2 or zone 3. I just finish up laser, because as they grow older, subsequent follow-up become pretty difficult, because baby won't cooperate. Probably sometime you have to put them under general anesthesia for their examination. So to avoid that, and if you have reasonably good normal vessels are grown to anterior zone 2 or 3, the remaining area, I feel comfortable. You do laser and you are done with. Otherwise, 40, 50, 60 weeks, 76 weeks, how long you are going to follow this? That's become a practical difficulty as well.
Thank you, Dr. Dr. Vishali. I would just come to Dr. Vishali now, because the main point that we discuss in each and every conference in terms of the immunogenicity. So my question to Dr. Vishali to you is that, for example, you see a lot of inflammatory, CNVMs and uveitis. Do you have any kind of reservation that patients who has uveitis, a drug which had a history of some kind of inflammation with few batches, would you have any kind of hesitation in use this drug in uveitis patients, for example, inflammatory CNVMs and all?
Not really, because to be very honest with you, we do not understand the drug completely. And we do not really know whether it is going to be more immunogenic in the patients who already have had some kind of uveitis, or it's just going to behave like any other drug. So to be very honest with you, that would not be my major concern, because I have so much in my armamentarium to control uveitis, and this little bit mild inflammation of few cells here and there caused by the drug do not bother me as a uveitis expert, because I can take care of them very well. But if it works for the inflammatory CNVM, and if affordability is an issue, and if the patients nowadays have a choice which one they wish to choose from, so I personally will not impose on the patient that biosimilar is non-inferior to biologic. It would be their choice, because little bit of inflammation to me is not a concern.
Okay, great. So I think you would offer all three as a...
Absolutely. That's what I do. I give them a choice and telling them everything and let them choose.
Dr. Alay.
Ashish, I think there is a very nice comment by Dr. Vikas Ketan, and what he has said is probably what we all echo, that probably the biosimilar ranibizumab in India is replacing Avastin. It's not actually replacing eccentrics. So if you see the surgeon's perspective, personally, I have shifted entirely from using Avastin to biosimilar. So I don't use Avastin anymore except in ROP.
We do not use Avastin at all in our institute.
Yeah, that's the thing. So
well, to add here what Ali said, because we have a lot of patients, almost 35 to 40% of free patients, I think we do not have a choice but to still use Avastin for this group of patients. We just cannot afford.
Can I just say something? I think in South India, we still use a lot of Avastin. I mean, at least in my institution, still 50% of patients who are getting anti-VEGF are getting Avastin.
Yeah.
So if I could make a comment as well.
Yeah. Dr. Cooperman wants to make a comment. Yeah.
So following up on what Vishali said, again, the amazing thing about these biosimilars, the one that's available in India, the Razumab, is its safety. That is, as we've learned from brolucizumab and abicipar, we've almost taken safety for granted. And again, it's fascinating. Abicipar, there was ascribed that inflammation to host cell impurities, but it was seen only in the first few injections and only in a subset of patients. So what I guess I asked for Vishali, do you think there, besides the host cell impurities, do you think there's an autoimmune component to that as well, that we're somehow a multifactorial process? Why only early, and why only in a small subset of patients?
You know, Dr. Cooperman, they did not have that much of occlusive vasculitis in their trial. And
Right.
they went back to the data and they scrutinized again, and it was not there. And what I have been thinking, maybe the trial had treatment naive patients. But when it comes to the real world, we are all very restless. If the patient is not responding to one and you want to switch, sometimes you do it in a period of a month or 6 weeks. You do not give that washout period of 3 months, which any trial would have. So I think there is a combination of factors, and to my mind, that immunogenicity between the two drugs may be contributing towards a component of occlusive vasculitis. But I think that is the scariest of all. You inject inflammation, to be very honest with you, 1-2 plus cells doesn't bother me.
Sure.
But occlusive vasculitis would scare the hell out of me.
I mean, brolucizumab as a new molecule, in a certain sense, a little bit of a bio better, not exactly. I mean, it was, again, looking at a fragment of a fragment of a fragment, right? We went from bevacizumab to ranibizumab to brolucizumab. They're each the subsequent, smaller components. So it's a brand new molecule, but then suddenly, unexpectedly, a small percentage of patients, the safety issue that we had taken for granted almost, we're so grateful for it. We, of course, are always worried about safety, but we stopped worrying because our drugs have been so safe. But brolucizumab, that looked like potentially the most effective of them all, had this rare or infrequent but devastating consequence. So, of course, it's largely shut down.
We have a paper coming, Dr. Cooperman, sorry.
Those. You know,
so
maybe
we have a paper coming. Sorry, Dr. Vishali, please go ahead. Please go ahead.
I was just talking to Dr. Cooperman, maybe do laser flare meter, OCT grading on vitreous cells, follow up and see what are the kind of the patients, that show worsening.
Right. We'd love to use it. It's an amazing molecule, but again, that risk gives us all pause. We are now using it only very selectively. Ashish, you were about to say something?
Yeah, actually, we have a paper coming in. If you look at the one paper that I showed, we had discussed this in 2019. We proposed the ADA component. ADA component is something which is not in your hand. So if you look at the brolucizumab and if you compare that with the aflibercept and the ranibizumab, ADA is the one which has the highest difference. You have almost like 50 times more ADAs, that to pre-injection in brolucizumab patients. So anyway, means that's a different topic altogether. So I would just come to Dr. Giridhar, because Giridhar and Dr. Savaran, they treat a lot of PCV patients. Do you have an experience, any kind of efficacy difference that you have seen with the originator and the biosimilar?
Comparing the biosimilar, we feel that the sub-RP component seems to respond a little bit better than the originator molecule. That's what we not had extensive study, but offhand I can say that some of the cases showed a better response as compared to the originator in drying up the sub-RP fluid on the height of the PED.
Yeah, Dr. George has shared that data at some place, so that's what I had that question. Dr. Girdhar, do you have any point to say on that?
Yeah, I think we have a similar experience to that the sub-RP element probably responds better with the racimab, with the biosimilar than the originator.
Any
but
other panels?
We do not have that many numbers
to
really come to some conclusion.
Right. Yeah, I think I agree.
Because in PCV now, as far as anti-VEGF is concerned, we are using more and more of aflibercept. So the number of patients who are using the other molecules are decreasing. So really, we do not have that many numbers to comment on that. But nonetheless, probably even Avastin actually in PCV, we find that the sub-RP effect is more than ranibizumab. Even that is bevacizumab. If you use bevacizumab for patients with PCV, the effect on the pigment, the sub-RP changes are much better than we compare with ranibizumab.
Dr. Cooperman, I think I have a question from you because that question comes right from the Biogen. Biogen US, and I think the entire team is watching the webinar. And they have some specific questions from you in terms of, I just read out the question. And Sushil has put out this question. You must be knowing Sushil. He's heading the worldwide medical for this Biogen.
Yes.
So
Sushil Panda.
question,
Yes.
So I would just read out the question. I would like to raise from Biogen that what are the 3 to 5 priorities to consider to choose a ranibizumab or biosimilar ranibizumab, which are going to come in US, or the original Lucentis. I'm not sure whether you got the question. They basically want to know the safety, efficacy, clinical endpoints, quality, history of the company, affordability. What 3 priorities you would see, for a company, which is trying to launch a drug in the US as a biosimilar?
I'm happy to address that, but I'm going to also turn it to Alexandra in a moment. So you put your thoughts together, Alexandra, as well. But it's a challenge. The United States is a challenge because we do have good compounding pharmacies, and Avastin at about $50 a dose or so is enshrined. So I think, ironically, the world's biggest market. I mean, the drug companies salivate over the United States because we're the ones that have happily or unhappily overpaid for drugs for decades. I mean, basically, the incredible price points in the United States have allowed the rest of the world to have lower price points, as expensive as it seems in India and in other places. Even Europe pays much less for their drugs than we do. Basically, America is subsidizing drug development for the whole world. And the system is tolerated it. But Avastin is an amazing end-around molecule. But certainly, I think what's been highlighted most recently is we have to show comparable efficacy, and it has to feel comparable. There's some chatter in our own chat line amongst the speakers about bio-betters and showing superiority versus just showing non-inferiority. It needs to shift from where we are. It needs to be very safe. Again, brolucizumab and abicipar have highlighted safety issues and has to feel as effective. And it doesn't hurt if, even though it doesn't show superiority, non-inferiority is acceptable, but if it has a sense that it does a better job. Many American doctors use treat and extend. It's a very popular mechanism worldwide. I'm not sure what the practice pattern is in India, but it's interesting that what we're always trying to do with treat and extend, of course, is to extend the interval. And there are many patients that still have fluid every 4 weeks. Why aflibercept became important or popular is we felt that we could extend some of those patients from 4 weeks to 5 week or 6 week interval, or they were drier at 4 weeks. Brolucizumab, until we saw the vasculitis issue, the hemorrhagic occlusive retinal vasculitis that terrifies us, the reports were that patients that were needing every 4 weeks of aflibercept suddenly were drying up. They could be extended further out. So even though there was no claim of superiority, if they dry better or if they have a sense that they're better, that is a way that a biosimilar can take into the marketplace. That is not the goal of a biosimilar, of course. A biosimilar is simply trying to do as good a job, as safe a job at a lower price point. Now, the question is, how much will that move the marketplace? Hard to know, because most people have insurance plus their supplemental insurance. But Alexander, what do you think? For example, let's make up a number. Would a $500 ranibizumab or a $500 aflibercept sway you? Again, Alexandra is in the mothership of Avastin use in America because her leader is Dan Martin, my dear friend, who was the leader of the CAT trial. So Avastin is enshrined there perhaps more than any other institution. What would it take to stop you to use Avastin, bevacizumab, and go to a intermediate price point biosimilar of some kind?
Yeah.
I think, Barry, those are great comments. I think we have several issues that are ongoing. I think safety is a huge issue these days. And as we've talked about, it's become more and more of an issue with the newer drugs that are coming to the market. And the awareness of safety is heightened. So whoever comes to United States, they have to show that it's safe. Because even if there is an inkling that it's not, I think the adoption is really going to be, is going to be hampered by that. Second is the cost. And I think that's going to be a challenge because, like we discussed, it's so expensive to develop a biosimilar, and it's not going to be Avastin. Now, I think it's different academic institutions versus private practices, because at our academic institution, our pharmacy compounds are Avastin, so we feel very comfortable with that. But there are still issues out there when you have compounding pharmacies. The final point I want to make is that for any company that is thinking of developing biosimilars and bringing it into the market, you have to make sure that there's education of providers and of patients, because patients and a lot of providers don't understand the difference between generic and biosimilar necessarily. And patients often come to us and they say, I was on this high blood pressure medication, it was working great, they changed me to generic, and then I'm having issues. And that's a common story. So educating the providers and the public about what biosimilars are is going to be very important as well. I have to admit, I think unless you're in this milieu, you might not realize what biosimilars are all about.
Okay, we have 5 minutes, so I will take some few quick questions. Dr. Harsha and Dr. Debulal, did you find any difference in terms of the efficacy of the drug for DME or other indications other than AMD?
We have submitted our the DME part of this for publication. We have certain differences, but these are not much. I mean, the efficacy wise, the drugs are neck and neck types. Yeah, neck and neck.
Dr. Harsha?
Yeah, we are using the drug. A biosimilar, we are getting the good result.
Okay. Dr. Raja, I'll just come back to you. You had, some kind of survey going on. Would you be, willing to share some detail in terms of that survey?
Yes. So, Ashish, thanks for organizing this meeting. I think it's very interesting. Can I share my slides, please? I'll just.
Yeah, that would be great. Yeah, please.
Yeah. So I hope you can see my slides here. Yeah, so first of all, I think you mentioned there are quite a few companies who are manufacturing biosimilars in India. I would like to just make a point based on data which we have got from the National Pharmaceutical Sales data. There are about 13 companies which I could go through, right from Dr. Reddy's to Alchemo, Hetero, and Reliance, and you name it, Mylan. There are so many companies who are manufacturing bevacizumab biosimilar. Right now, there are quite a few other ranibizumab molecules which are going to come in the market now. But if you look at the overall market share of Roche's Avastin versus other biosimilar bevacizumab, currently, I would say 30% of the market share based on sales data is held by Roche's Avastin. And the remaining 70% is with the biosimilar companies, all the others put together, 12 or 13 companies put together, and it's close to about a $6 million market or so per month is what I'm conveying here. So overall, I would say that the biosimilar market in India has picked up significantly based on at least the bevacizumab biosimilar data. And this graph slide over here shows that 5% of retina specialists way back in 2017 were using ranibizumab biosimilar, but by 2020 you can see about 90% of retina specialists have used both ranibizumab and bevacizumab put together, but very few have used only bevacizumab biosimilar. And the preference is not for bevacizumab biosimilar. So we did a survey recently about on diabetic macular edema, and you can see now this is 2020. In 2017, it was 5% for razumab biosimilar. Now it's 20% of the retina specialists are routinely using razumab biosimilar. As has already been mentioned, razumab is eating into the market share of Avastin instead of ranibizumab, Eccentrix, or Lucentis. And aflibercept has not picked up significantly, although in PCV patients, sometimes we are going in for aflibercept Eylea, but again, because of cost constraints, it's not picked up significantly. But here's a point for INTAS, if they are watching carefully, based on the 2018 survey and 2020 survey, the efficacy analysis of doctors perceiving the efficacy of ranibizumab biosimilar, that has gone up. They feel that it's very, very efficacious compared to, let's say, Lucentis or Excentrix. But if you look at the safety, while the yes numbers have also gone up, the no numbers have also gone up, in the sense there were quite a few in 2018 who did not respond to that particular question because they had not used it probably. But in 2020, the number of people raising concern about ranibizumab biosimilar has also increased. So that's something they should keep in mind, and probably standardize their processes so that there are no more adverse events happening in the future. And someone had asked a question about bevacizumab biosimilar. This is just a graph to show that most of the retina specialists have not used bevacizumab biosimilar, but among those who have used, 30% were not satisfied with bevacizumab biosimilar. They would probably prefer Avastin over bevacizumab biosimilar. We don't know, but a significant percent of retina specialists who use bevacizumab biosimilar were unhappy with that. And this is just a graph. Probably Intas would know much better than I know about this. This is the sales data of Razumab biosimilar. And because of the adverse events, you see a dip in 2017, then it picked up again, then again there was a dip in 2018 because of the adverse events. So overall, I would say these are based on survey analysis. And overall, I would say Razumab has picked up from 5% to 20%, but it's eating into Avastin share, as has been rightly pointed out.
I still have questions, but I think
Ashish, can I just ask one question to the
Please, Dr. Girdhar, if with the permission of everyone.
I mean, the thing is, I mean, all agree. I think. See, the question is not about this efficacy of the biosimilar now. I think the biosimilar is quite effective, but the main issue is the safety. So basically, I think it will be interesting if we get a pool data from of the Razumab data from India. For the last 3 years, it will be very good information. We only look into the safety, nothing else. How many patients had inflammation, how much was having the gradation of inflammation, how many of them had permanent visual loss because of the drug, etc. If we can do one sort of a safety study as far as Razumab is concerned, I think that will improve. I mean, that will improve our confidence level.
Dr. Vishal,
even today, I am little reluctant to use it in one-eyed patients, in patients with very good initial visual acuity, etc.
Dr. Vishal has some point to make.
So it will be very interesting if we can get some sort of a data like that. And I mean, if we can work, if you can take an initiative on that this thing and start doing something on that count. I don't know whether Pramod will agree on that. Because these sort of 100 eyes, 200 eyes, really doesn't. I mean, when you have few thousand eyes, it gives much more power.
Sir, I think what we have as per the ADRs, if you look at my slide, means they have approximately 130,000 vials till now, which are being in the market. So whatever ADRs were reported, I have actually categorized them as per the year. So they were actually quite low. I'm not sure, because whether under current, not reporting data, I'm not sure, but reported data was quite low. So that we have. Dr. Vishali has a point, please.
Intas has done a trial in India already, and they are in the process of publication, the results of the Indian group. So I'm sure it will come out soon to what Dr. Giridhar is saying. The safety profile was quite acceptable in the trial.
So I think I still have. Sorry.
One quick point. I know that we're running out of time, but one thing that's important to note about safety, and back to Dr. Girdhar's question of pooling the data, is one of the things why we like the established drug company, the established originator molecule, is the consistency from batch to batch. The processes are so well established that there's not a lot of variability. When there is suddenly variability, as there has been intermittently with the aflibercept, it raises a lot of concerns. So as the biosimilars roll out with smaller companies still with huge expenses, the concern about pooling data, Dr. Girdhar, is that maybe you're looking at the different batches and And as their processes improve, we've learned with abicipar and brolucizumab, they're working even on those new molecules, trying to make them safer by improving the manufacturing process. It's not the molecule they don't perceive as being unsafe, it's the manufacturing process. I think we need to be aware of that. From company to company and from batch to batch within the company, there may be some variability.
I agree with Dr. Cooperman actually, but what happened with, I think what looking at INTA's group, what happened with Razumab, in between those couple of batches and when they learn their lesson with the stringent guidelines, after that, there's hardly any issue. And I think they are fairly consistent now actually.
Right.
And we do not have any hesitation using any patient, whether it is one-eyed or otherwise, did not have any problem at all after that. Yes, initially, I think they learned their lesson well.
And I think that's a good point. That's why I wouldn't pool the data necessarily, because you may be clouding the waters, so to speak. It may be important to watch the progression. So while you can pool it, I think it's important to, while you aggregate the data, you also have to subset the data and look at it over time potentially.
See, published data with the available biosimilar is very, very limited. See, that's where the whole issue is, as Barry said.
I would just touch upon Dr. Girda's question, I think, to Dr. Cooperman. You know, there's always a question raised on biosimilars that you had less patients in the clinical trial. So, you know, that's what the question that we have been asking in a lot of forums. So do you have any kind of, stake on that?
Well, conceptually, a biosimilar shouldn't need to have as extensive clinical trials. That would undercut the purpose of it and the development costs. It would make it be a more expensive molecule. So be careful what you wish for, right? The more studies means the more expense by the company, and therefore the higher the price point to make their money back. So generics don't even need to do any clinical trials. They just need to show equivalence of a molecule, and the vehicle is close enough, show certain things with very limited clinical trials. Biosimilars, by definition, need some clinical trials looking for safety and efficacy, but they need to have at least one good trial for one indication, but don't have to necessarily show up for all the indications the way that an originator molecule has to have individual trials for each indication.
I think I still have a lot of questions, but I think we are running out of time, and everybody has something scheduled. So thank you so much, each and everyone. And I think the take-home message is safety is as a paramount importance, and efficacy. We all know that the drug is good in efficacy, but I think safety is the one which all the drug companies which are planning to bring biosimilars should take care of that, the way they should monitor it. And I think I can't thank enough to all our overseas speakers and Indian speakers for sparing their time. And I think we still had a lot of questions, but we should, in respect of time, we should stop it here. Thank you so much, and have a good night, and have a great day.
Thank you very much. Thanks for having us. Thank you so much. Good.
Very nice. Very nice.